Good afternoon, everyone. I see people still joining in, so let's take a moment to settle down. All right, so probably let's get started. On behalf of the ImmPort team, I extend a warm welcome to everyone. We appreciate you being here, taking the time to join us today. My name is Sanchita Bhattacharya. I'm the scientific program lead for ImmPort project, and I'll be moderating the session today. And with me are Kate Hives, Sammy Smith, and Matthew Strubb from ImmPort team, and they will help me in running this session. So I will start with giving a brief overview of ImmPort, those who are new to this project. I'll just quickly go over some of the features of ImmPort. And ImmPort is immunology database and analysis portal. It's a data sharing platform funded by NIAID, National Institute of Allergy and Infectious Diseases. ImmPort ecosystem has private data space, shared data, through which all the public data sets gets disseminated. We also have data resources available or for tutorials, documentations, and some of the data analysis tools as well. And we are proudly kind of doing a fair sharing of the public data sets. And so with that, the question is like, who shares the data through ImmPort? And so ImmPort data primarily shared for the major NIAID funded programs, and we also accept data from external organizations. The list is pretty long, but I will just highlight like HIPC, the Human Immunology Project Consortium, Seronet, and IMPACT, submitting data on COVID, and Civics for the vaccine data sets, much of DIMES for preterm birth. We also have autoimmune disease data sets in ImmPort, some of the tuberculosis data from Bill and Melinda Gates Foundation, and on the right-hand side, you see a long list of many other programs submitting data. And it's our 20 years of data sharing in the space of immunology. How do I access data? So ImmPort has built an interface, ImmPort shared data browser called Cohort Discovery Tool. So ImmPort currently shares over 1,000 studies encompassing a range of research areas, autoimmune, vaccine response, immune response data, and then transplantation data, and many others. We also support 185 clinical trial data getting shared. If you are interested to explore this interface, please feel free to scan the barcode, and that will take you to this website, and you can search your data sets of interest. So now we move on to the main presentation, and I would like to cover a few housekeeping items. The presentation time is 45 minutes, followed by 10 minutes for Q&A. And please feel free to post your questions on the Q&A window at any time during the session. And we will also take few questions live after the presentation. The webinar will be recorded and made available for future access. So with that, without further ado, it is my pleasure to introduce our distinguished speaker, Dr. Afshin Bhashadi. Dr. Bhashadi is affiliated with many prestigious research institutions, including McGowan Institute for Regenerative Medicine, Center for Space Biomedicine at the University of Pittsburgh, the Blue Marble Space Institute of Science, Broad Institute, and many others. Today, Dr. Beshati will be presenting the research he recently published titled "To Boldly Go Where the MicroRNAs Have Gone Before, Space Flight Impact on Risk for Small for Gestational Age Infants." And so with that, and his title today is Harnessing NIAID's ImmPort and NASA GeneLab through Computational and Systems Biology Approach, Determining Female Health Risk for Space Flight Driven by microRNAs. So let's welcome Dr. Beshati. And while he is getting ready, I just want to give a big shout out to the study, because this study is a part of an active collaboration between the Radiation and Nuclear Countermeasure Program Group, Dr. Carmen Rios and David Cassat, and the Biomedical Informatics Group, Dr. Dawelin and Dr. Anugaraj, with NASA GeneLab members, and the datasets provided by Machu Dimes, which are powered by ImmPort in GeneLab. So with that, I'll stop sharing and turn it over to Dr. Beshati.
Yeah, thanks. Yeah, thanks for inviting me here to give the talk. Let me load up the talk. Okay. So yeah, now thanks for having me here. Thanks everyone for listening, and I look forward to seeing what everyone has questions. And as I go through my talk, keep in mind, as so a lot of people might know me or don't know me, that I'm very open for collaborations, and how I work is the more the merrier approach. So if people are seeing things that potentially collaborate in the future, reach out to me. And the top email here is probably the best. Reaching out to me, and that'd be great. So then you could talk to me and figure out ways we could work on various new projects that are related. So I do a lot of different work, and the central theme for a lot of my work is mitochondrial related work and microRNAs. And today the focus mainly will be on microRNAs. So keep that in. And then I use, of course, systems biology approaches. And until I said briefly, I have an open science approach, which is very, very much in line with ImmPort and GeneLab, which is now called the Open Science Data Repository, and the large network of people I work with from around the world, which I want to keep expanding. So the area that I'll be working on today is on space biology research, which I have lots of work done on previously related to microRNAs, mitochondrial, and other type of work. Before I go on too, so some people who don't know me, I'll give a little background. I joined University of Pittsburgh on September 1st. And I've been here for now almost 2 months, or a little over 2 months. And one of the reasons I came here is to form a Center for Space Biomedicine. So we launched this Pitt Space Initiative about a month ago. There'll be 3 other components, where there's a big space engineering component here at University of Pittsburgh at Pitt, and space science, and then I'm leading the space biomedicine. And the whole kind of pillars of the space biomedicine program will be involved in these core topics, and more to add in the future. And which includes, female biology and the reproductive system, and the sublayers. Once you hit these sublayers, well, I'll include that. But and lots of other types of research. So if people want to collaborate with me here at Pitt, we have a lot of faculty interested in working on space biomedicine. That's another avenue where you could partner with us at University of Pittsburgh to actually work on nice space-related questions. And one other thing that people might be interested in, and even people at ImmPort and at NIAID might want to collaborate with me on this project, is an education outreach program that I'm wanting to do. So the goal is I want to do a global education certificate and eventually a biomedicine programs degree program, where Pitt's the hub. But the idea is, people are teaching space biomedicine isolated in their universities or other places. Some universities do have a space biomedicine degree, but most don't. But the thing is, I have my expertise, but their expertise, I don't have expertise in an entire field of space biomedicine. I know about them, but why not bring our friends and colleagues who know about that specific topic and expertise to teach that course? So basically harmonize the education program for space biomedicine across the entire world, where the idea would be that one course is provided for all universities, and then each university will have their credit, how they process the credits within their own university. But for example, one course could be on space radiation, and it'll be taught, let's say, the first day here at Pitt by me, and the next day simultaneously by one of our collaborators, let's say at Harvard or at Oxford or one of our other partners from other universities that are signed up to be part of this. And so every student's getting the same exact course, same exact teachers, and same exact curriculum, no matter where they are in the world. And then through their university, that the university can work out how they get credited for that. And so this harmonizes space radiation, and then has all these kind of core ideas, global effort. We also try to include some of the GeneLab for University program there. They learn about that, and hopefully maybe ImmPort might be interested to partner with us, because then they could learn about how to use, because space, and I'll tell you in a few slides down, space is an accelerator model for a lot of diseases, including aging and including infectious diseases and other types of immunological diseases that you guys might be of interest. So, this might be a kind of a collaborative partner with folks here. And so right now there's a lot of interested parties. There's actually more universities and agencies involved. There's some nonprofits that are and charities involved. So some for-profit companies is more actually that signed up. And the idea, let's say for-profit companies or space companies that sign up, is now they can actually know the curriculum the students are learning. And then obviously they could know exactly which students they want to take for their company straight out of, once they graduate, because now they know they've gotten the exact educational course that they need for their companies. And then also all these universities are involved. We know exactly what the students are getting, because no matter where you are, both NASA and European Space Agency are interested in the project and how they can help them. We'll talk further. But one idea that was presented by someone at NASA was, why don't we create a space biomedicine textbook? And that's a great idea, where we could be core editors, but then people from the community who want to be involved, we create the standardized space biomedicine textbook, just like every field. Like in physics, we have our magnetic field textbook. That's this big red textbook when I was in grad school that everyone knows has done it. So we're trying to create fundamental. Cell Press is interested to somehow partners. Maybe they might be the partner for that. But that's just throwing this out there. Anyone listening, they can be involved. Reach out to me. The more the merrier, because if more people sign up, that means space biomedicine will now be harmonized, and everyone will get the same exact teaching, and more people who with their expertise can sign up to be involved. Okay, so that was the little commercial part of it, saying what I do, what potential aspects we could do, and what other people could maybe help with. But now, why care about research in space? I mean, a lot of people here might not have done space research. There are probably some people here who have. But one thing to keep in mind, space, as I mentioned, is a cellular model for aging. So you don't age quicker, but the phenotype and disease that set in that are associated with it happen a lot, tend to set in quicker, and the onset of it is a lot quicker due to well, it's just showing a few slides down, physical hazards that happen in space. One of it is also mitochondrial diseases, which I won't get into as much this talk. So we kind of dubbed it, me and some colleagues like Chris Mason have dubbed this as the second space age, meaning, if you look at how many samples are actually being launched to space, pretty much from the beginning of the space launches in the 1960s until pretty much the last 5 years, it's been a steady flow of things. But you can see how since 2020, mid 2020, a little earlier than that, there's been a huge explosion of things launched to space, and not just humans and biological payloads, but also satellites and other things. And this is due to the fact that before it was just like a race between two countries, right? But now there's so many other players involved, not just the US and USSR, Russia now, but a bunch of other countries from Europe, from Asia, and so on. And now all the commercial companies like SpaceX, Blue Origin, and Virgin Atlantic, and so on and so on, that are involved. And the next obviously goal is to go to set up a base on the moon and eventually go to Mars and beyond. So I mean, that's why there's so much importance, is the moon and then eventually Mars. So to do all this and make it safe to travel in humans in space, there's 5 hazards that NASA had defined that we need to overcome, so be really safe for humans to travel. One is the distance from Earth. Obviously you have limited healthcare and support. The confinement, I kind of think it should be only 4 hazards. Trying to start a campaign for this, but the 5 hazards, because confinement and hostile and closed environment are sort of the same thing. But confinement means you're stuck in a tube, so your lack of fitness. Hostile closed environment means in that closed environment, things like infection and things are a lot easier to obtain between individuals and get from your environment. And so those are some things. But 2 of the main things that I'm going to concentrate on of the health risks is the lack of gravity or microgravity in the radiation, meaning specifically space radiation. And of course, to figure out these things, we do experiments related to samples sent to space, and I'll go on this in a second, or ground simulated experiments using not only samples from humans, since those are limited, but also using lots of commercial type, not commercial, but lots of other organisms, from mice to fruit flies to fish and so on, microbe cells, and eventually using computational tools and analysis and data analysis to figure out what health risks are happening in space. And then eventually the ultimate goal is, what's the magic pill to make it safe to travel in space, or the countermeasure that's needed. I mean, currently, yes, astronauts do exercise, but that's obviously not enough to mitigate all the damage caused. It helps, but it's not enough. So the space environment, as mentioned, there's the space radiation, and then there's the lack of gravity, microgravity. So I'm gonna go in a second what space radiation is. You got two different types. You got the solar particle events, which is one high acute dose of protons, as you see there, at different energies. Usually, that's coming, so you might be able to try to reduce the dose ahead of time by mitigating somehow. Obviously, it's not enough to fully stop the damage caused by this type of radiation, but this doesn't happen as often. The main type of radiation is the galactic cosmic rays, which is the background radiation. And the majority of the ions in the background are protons, as you have here. And eventually you get the iron particles. In Earth's atmosphere, or not atmosphere, but orbit, you got the Earth's magnetic field. This is the Van Allen belt. And the magnetic field itself, due to the physics, will reduce the doses that you get in deep space. So you got to keep that in mind. So all the experiments that are currently or majority of it being done, and the data available is from the ISS, the International Space Station, is within the low Earth orbit, and getting the astronauts reduced to a lower dose. So the Fantastic Four, they thought back when this first came out, if you get exposed to galactic cosmic rays, you're going to get superpowers. So let's see if this is true. So one thing you got to keep in mind is that what we refer to in radiation biology, and the type of radiation, is LET, which is referring to linear energy transfer. So this is the amount of energy an ion transfers to a cell or a tissue. So everything you're exposed to on Earth, like gamma radiation, X-rays, and so on, it's low LET radiation, meaning particles that travel very fast. It goes right through your cell like grains of sand, and the energy as it goes through your cell is deposited. Now, the heavy ions you see in space, as I mentioned, protons, the iron particles, mainly. Once in a while, you get a heavier particle, but the majority of particles lie here, are the high LET, meaning these are particles that move a lot slower, but it's like a bowling ball. Eventually, the energy is deposited right in your cell, and it will cause a lot of damage and do a lot more effects and increase health risks, as opposed to the type of radiation you get on Earth. And so let's say if you compared to types of radiation that are the similar doses, the millisieverts here. So if you have, let's say, the gold or yeah, these are probably gold, yeah, color ones. Let's say if you have two doses of a, let's say, 7-day-old shuttle mission in the low Earth orbit, is about 15 millisieverts the astronauts are exposed to, but a CT whole scan is almost the same. You might say, oh, they get the same radiation, but remember, I said this is a lot more damaging. So there's a terminology called RBE, relative biological effectiveness, which compares them comparable damage caused by radiation here compared to this type of radiation, the low LET. So going to Mars and coming back is like estimated you'll get exposed to a total dose of half a gray of radiation, this galactic cosmic ray. Now, or let's say if your 7-day shuttle mission is about a 15 millisieverts of, in the low Earth orbit, which is almost equivalent to a CT dose. But you got to keep in mind, this dose, the damage caused to your body, is about 10 times more damaging than this 20 millisieverts you get in a CT scan. So this is really like more equivalent to maximum core exposure on Earth, which is this background radiation you see in this place in Iran. So that's what you got to keep in mind. The damage caused by anything you see, the radiation doses you see in space, is at least 5 to 10 times more damaging in space. So in reality, I think the Fantastic Four, the amounts of radiation they got, it would have been only one issue comic, or they would have just been in the hospital. So they might have to go back to Marvel and tell them to rethink this. Okay, so then the space environment, all this damage caused to your body, what does it do? You can imagine it causes a lot of health risks. So the health risks there's some female and male independent type health risks, and then there's some universal type health risks that happen independent of sex. So and then in this case, I'll be talking more about the female reproductive system, so the health risks associated with that. But there's all these, and of course, the longer it makes sense, the longer you're in space, the more potential you have of the health risks increasing. So you have everything from renal kidney issues, and even this thing called space-associated neuroocular syndrome, SANS, which the astronauts seem to start losing their vision. You got liver issues, you got sleep disorders, so on cancer risks, immune disorders, metabolic disorders, and so on and so on. So you got to keep those all in mind, is basically every health risk under the sun. So if there's any other ImmPort data, for example, that you think of, because there's so many different health risks associated with space travel and a lot of immune-related functions, and think of how to relate it. Are these things similar to what might be seen in space? We should collaborate, because there's a lot of different stuff we could work on with your expertise here. So what type of experiments can you do? You could actually launch mice into space. So these are mousestronauts, but of course, not everyone's capable of doing that and have the funds to do that. You can see the mice eventually have a good time, and until they're unfortunately reach the end point of the experiment. But they have a good time, and they tend to whiz around the cage and not be impeded by them, or adapt to the lack of gravity fairly well. Now, as I said, not everyone has a chance to launch things to space. You could go to a place called Brookhaven National Lab, which is a high energy physics collider in Long Island, New York. And NASA had, back in the early 2000s, set up a NASA Space Radiation Laboratory there, where you could take, in the high energy physics colliders, you could produce those heavy ions from protons to iron particles. So they set up a laboratory there, the beam room, where you could actually put mice or cells or anything you want in front of the beam and expose them to the heavy ions and simulate the space radiation you might get in space. So that's one way of doing simulate experiments on the ground. And another way to get the microgravity simulate experiments is hanging the mice from this. It's kind of silly, but it's called hind limb unloading, where you hang the mice by the tail. So it's basically unloading the weight from the back leg, causing the muscle generation or weight or muscle reduction use that you might see in space, and the kind of the fluid shifts you might see. It's not true microgravity. You can never truly simulate microgravity on Earth, but as humans, we try to get somewhat close and try to have a similar behavior. But so this sort of gets to that. And again, for space radiation, keep in mind that the doses we give is usually an acute dose. That's the total amount of dose that the astronaut might be exposed to over the year-long mission, for example. So in space, you're getting a constant lower dose over time, every day. Like basically over a year, if you're in space, it's been estimated, every cell in your body will be at least hit by one of these ions that are in space. So you got to keep that in mind. So the biology might not be the same, but this is the closest we get to simulate. The idea is if you use the total accumulated dose, so let's say if you go to Mars and come back, and if you're able to mitigate the damage caused by this high dose radiation, most likely your strategy for your countermeasures should work for a lot lower dosages you get over time. So, and there's other kinds of gravity models that are microgravity models that, let's say, Stuart Rucker and True had made up. It's called the partial weight bearing rat models that you could use. And other types of experiments that you could also find on available publicly data, which I'll go over in a second. You could put worms people have done, zebrafish, fruit flies. Here's the fruit flies. The plants, there's a lot of plant studies that people are looking at, microbe studies, and fungi studies. So there's a lot of different things people work on. People have even sent up squids. Each of, as you know, each of these animal models represents some certain feature of the human physiology. So if you have any favorite animal models that haven't been sent up in space, maybe we could apply for NASA grants and see if they can send it up. And of course, just like ImmPort, NASA has set up this. It used to be called GeneLab, that's the old kind of platform there. And now, initially when it was set up, it was all just omics data, and where all space-related omics data appears. But now it's the NASA Open Science Data Repository. I should put the newer website picture up now. But the Open Science Data Repository now just doesn't have omics, but it has a whole bunch of tons of other data from just kind of like ImmPort, but our ImmPort has lots of different types of data, from imaging to Westerners, proteomics, and omics was before, but a bunch of other things, non-omics related data that's on there now. So you could go here and let's say, search for your favorite tissue, favorite omics data you want, and then see what type of data is available here. There's lots of data there. And then one other thing, there's the new open science data I have here in this slide for the picture. But one other thing that people can sign up for is GeneLab, when first formed, we set up these analysis working groups And originally it was 4 groups, but now there's a lot more. I lead the multi-omics, this is biology multi-omics group. That's been around since, I guess, 2018. It's that long, so that's a long time. So, but then what we do originally was set up so, you know, the community come together and come up with a consensus on how standardized pipeline to analyze the omics tool. But obviously, when you get a bunch of scientists together, they want to produce some nice, fun science, and this is what we've done. So with the multi-omics groups, we've put out a lot of nice papers and worked on many projects, and we're working on many more projects. So if people want to join that's another way to work with the community and put out some really fun projects with ImmPort, possibly data. Okay, and lots of papers were produced. People want to know about space biology, so I'm going to get to the paper in a second. People want when's the paper coming? But there's this paper cell package that me and Chris Mason and Susan Billy organized with Cell Press back in 2020 that came out a bunch of papers. This is a paper that was kind of centralized for the mitochondrial dysfunction. Again, a paper reduced from the analysis working groups lead. And then most recently, you might have heard back in June this year, Chris and I worked with Nature Portfolio to release a big, large package of papers surrounding this first all-civilian mission, Inspiration 4. So one thing to keep in mind, because you're going to see some Inspiration 4 data related in the paper that we put out, is that the ISS is 420 kilometers above Earth's elevation, still in the low Earth orbit, right? But the Inspiration 4 went up to 590 kilometers, which is still in the low Earth orbit. But as you get closer and closer to the edge of the magnetic field, you're going to get less protection from it, and you're going to get a higher increased dose of radiation. So these crew here that were in space for 3 days receive equivalent to like 6 to 9 months of radiation if you're on the ISS. But all the data that was produced from him, Chris and his team produced, and some other PIs produced lots of omics data and lots of data points. And then now all that data is available publicly on the Open Science Data Repository. So go there and find it. And this is the first ever human data to that level is available, anyone from stuff in space. So that's exciting. But also there's lots of papers like about that the community we put out together. 22 of them are from Chris and I, or a little more. And that which had data included for the Inspiration 4. So take a look at that. And although this paper I'm going to talk about came out after this package, it does include data from this mission. And there's lots of papers. And then, of course, we put out this paper kind of as the overall umbrella paper we call it for the entire paper, describing all the papers, and then we got the cover Nature, talking about kind of what's the relevance of the second space age and why all these things are important. And of course, if you don't know, why care about space other than the facts that I've told you about the accelerated model of aging, accelerated model. So everything you can do can apply to the clinical aspect. If you cure something in space, you definitely can cure that health risk on the clinic. But also the second space age is going to be a really multi-trillion-dollar, it already is, but it's going to be booming more for industry and people to be part of. All right, so the question that was proposed for us was, can we leverage publicly two publicly available data sets, meaning ImmPort, with GeneLab, and come up with a novel hypothesis that could advance both clinical and spaceflight issues? Well, I'll say yes, because I'm going to show you why. So short answer, yes, and I don't have to go into what ImmPort is, because everyone here obviously knows what ImmPort is. So I'll skip this part. The other part is I just told you about Open Science Data Repository for me called GeneLab. So yes, so let's see what we do if we can combine this. So the data set that, I mean, everyone has their key expertise, and of course me, loving microRNAs, I said I'm going to work on see if ImmPort has any microRNA data sets. And indeed, there's this very nice data set that I think some of the authors from this paper might be here in the audience, but so they had this very nice data set on maternal plasma from the microRNAs, and the microRNAs that were related to this specific birth defect called small-for-gestational-age, which is an unfortunate birth defect that, for short is SGA, that when children with this defect is born, they're smaller, of course, size and proportion than normal size children for birth. Some of these now children recover as they grow and they come back to normal, but unfortunately some kids don't, and they have some health effects because of it. So this group here put out this really nice paper working on how, let's say, the microRNAs during the gestational periods might be predictive or might show potentially why this could predict, let's say, if you're going to get this potential risk of SGA or not. So this was nice for that. So then the question that you would impose is, or I would impose is, and remember I said that or maybe I didn't say, is that female reproductive system has been not as well researched in space research. There's been some great groups and people working on it, but it hasn't been as researched as the more popular diseases and health risks that are out there, or the more common, like cardiovascular disease or cancer risk and so on. So the question I asked is, are microRNAs associated with SGA that we see here, for example, in this paper, and the data that I'm going to pull out from this paper news, can they be seen to be increased for spaceflight? And if these things are maintained after, the females come back for spaceflight, and let's say a female decides to have a kid and the microRNAs are expressed, can this potentially increase the risk of birth defects, So obviously, if these microRNAs that you have here are elevated during pregnancy, that's going to be bad news. So if you go into space and these microRNAs are elevated during when they're coming back, and they are actually still elevated when they come back, that's of course going to be bad news for them during birth. So what are microRNAs? Hopefully most people here know about this, so I'll go through this quickly. So microRNAs are one type of RNA, non-coding RNA, and then non-coding RNA are RNA that don't make proteins. And usually they don't travel alone, or they never travel alone. They have this protein attached to them called the Argonaut, which protects them from degrading and helps them get in and out of cells. And so microRNAs are actually very stable because of this. And classically, before they were discovered, this is sort of the classical view of biology: DNA, translated RNA, translated protein. But once microRNA is discovered, as we know, the Nobel Prize was given for the discovery of microRNA by Victor Ambros and Gary Ruvkin. But when they first discovered it, these small RNA previously people thought were just garbage and debris, but indeed they were not. They're involved with every aspect of biology. And one microRNA can actually bind through this 8-base seeding region to gene, and could bind to hundreds or thousands of genes. So they could regulate lots of pathways, right? And due to this size, again, I said their stability can be found floating freely in many parts of your body. Every bodily fluid that exists, it's there. MicroRNAs there. They're not only, they're involved with a lot of healthy functions, but they're also involved to be known for all aspects of disease. And they're not only found in mammals, but every living organism, from plants, microbes, and so on, have microRNAs. And one interesting thing is that microRNAs are highly conserved between species, so especially between mice and humans. So if you see the pattern microRNA that's happening in mice, most likely that's microRNA will be well conserved with humans, and it will translate from mice to human very nicely. So if it's a floating microRNA signature, that could be a potential biomarker for health risks or disease, and also potential therapeutic for therapy for diseases, targets for therapy. So I'm a systems biologist, physicist by background. So my view of microRNAs is this way. So a lot of people might have said, "Oh, microRNAs are so hard to deal with." When you actually, let's say, work on an experimental condition versus a control, you're going to get thousands of dysregulated microRNAs. So what's the most important? So how I look at it is that, let's say classically, one person per people might have looked at one microRNA at a time. For example, this LET7A is one of the first microRNAs discovered, and all of this big hairball around it is all the genes that it binds to. So if you just look at this one microRNA, say in cancer, for example, it's thought to be a tumor suppressor, so it's going to decrease health risk. Now, if you look at 2 microRNAs at a time in a setting, and one microRNA, like this is miR21, that's, let's say, a cancer promoter in this example, and it has equal influence on these hairballs, but they have equal influence on the genes, you might not see any health risk due to the balance you might see. But it's really never 1 or 2 microRNAs. It's a group of microRNAs that's causing the change that happens with a disease, or in this case, an insult of injury to the body of space. So it's the balance of these microRNAs causing the decrease or increase of health risks. And the other part is, this is a tumor example here again. The tumor microenvironment and the environment around there, it's important, but whenever there's some kind of disease, it's not just the immediate surrounding environment, but it's the actual systemic response in your body. So what would cause that to cause a systemic response? And as I said, my hypothesis would be the microRNAs. So if you figure out the right microRNA signature, which I think I have a pipeline to do that, then you can find a microsignature associated with every disease that's related to that to predict the health risk. So that's my view of microRNAs and how to move forward with it. What I've done so far, I'm not going to go through any of these papers, but I've identified a microRNA signature related with spaceflight damage to the body. In this paper, we show the original paper, we kind of confirmed that this microRNA signature exists in simulated mouse experiments and from even human astronaut data from the NASA twin study data that had microRNA sequencing done. And then what we had done is started saying can you mitigate the damage caused by inhibiting some of these microRNAs? So this 13 microRNA signature, we said 3 of these out of that microRNA signature associated cardiovascular risk. So our collaborator Pete out of Columbia University, he had this microvasculature 3D tissue model that we irradiated. And if with radiation, they'll collapse, the vessels collapse, but then we gave an antagomer, basically antagonist sequence therapy for those 3 microns, and we completely mitigated the damage caused. And indeed, and then the decadal survey, National Academy Decadal Survey, which is this 10 years, decadal, hence decadal, but which is this 10-year basically report that comes out every year on space-related research from the National Academy of Science that would guide the next 10 years research. They talk about how important microRNAs are. So in this paper here, this was part of the recent package, we actually further dive to what the biology changes are. And indeed, like overall globally, the gene changes come back to normal, closer to normal. And we talk about how inflammatory signals are reduced, if DNA repair efficiency is improved, and mitochondrial function is improved due to the inhibiting of the 3 microRNAs and rescuing the events. So this is an example of, if you know the right microRNA signature, you could potentially prevent the damage caused by the let's say space radiation impact. So in this paper now, we're actually looking at the potential birth defects caused or microRNAs associated with this potential birth defect, as I said. So this is the paper that maybe some of you have already seen that came out almost a month ago now. And again, it's a large group of people working on it. And actually, I was kind of impressed that they let me keep one of my silly titles. Usually editors tell me to remove the silliness in the title, but I was happy that this one snuck through. So the data sets to relate now to that nice import data set that was there is on GeneLab. I was like, is there a microRNA data set? And I said, of course there is, because this was a data set that I actually added to Open Science at GeneLab back then. So it's from my own experiments. So I knew that data set's there. As I said, I'm a little biased for microRNA research. So this was from an experiment done from the simulated mice radiation and exposure to space environment experiments at Brookhaven National Lab that I mentioned earlier that you can do. So there were mice that were hind limb set up for the microgravity, or mice that were normal, so they didn't have microgravity conditions. And they were done 2 weeks in that condition, because usually to get the onset of the microgravity impact, you want at least 2 weeks for them to be set up hind limb to get the full effect of the microgravity simulated, in quotes. And then we irradiated the mice with the galactic cosmic array. We had also the solar particle event. That's I remember like simulate the solar flares. And then we had a gamma reference, because sometimes you just want to do a gamma low LET radiation references, compare what the terrestrial response would be. And then the day after radiation, we sacrificed. So this is immediate time point. We didn't look at longer time points afterwards. And then there's another data sets of cells that we use, but this is not as important as this is now. These are female mice, so this is similar. These mice were not pregnant, and the whole point of the study is we're not going to launch pregnant females to space. We're going to launch normal, non-pregnant females to space. They're going over to space. The astronauts, same with the mice. So if they come back and they still have elevated microRNAs, then that's the potential if they want to give birth for the microRNAs to stay elevated. So just looking at the global responses of the microRNAs happening, no specific microRNAs. This is like PCA plots looking at changes. The top is the import. This is the import data sets here. One thing is the import data set from the paper, they looked at 3 different gestational periods. What eventually I did is grouped the gestational periods together, and there's differences happening between the gestational periods, but there's a distinct pattern of microRNA signature different between the controls and the SGA group. And the whole goal of why I wanted to look at the microRNA as a whole as opposed to the different gestational periods is that I want to look at that microRNA signature that's constantly being expressed over just time points A, B, C for the gestational period. That way, those are the microRNAs that consistently might be involved with SGA, as opposed to some microRNAs that might pop up later during the gestational period. So the first time point A would be equivalent to, let's say if the females that came back from space, they still have these microRNAs elevated in their bloods, and then time point A would be those microRNAs that would be potentially from space travel being elevated in your circulation. And then again, this is for my simulated space experiments. So one thing is then you can run some pathway analysis on microRNA sequencing data, similar to gene set enrichment analysis, if you guys are familiar with, where you could throw in your gene expression. So the pathway analysis, briefly, the tool you can use in our programming is that you put in the microRNA expression data, and then what it does, it works backward, because in microRNAs, as I said, they know the gene targets for the microRNA, so it works backward to figure out the up and down microRNAs, how the genes it targets, meaning the genes that would be suppressed, then it'll figure out that indeed, this pathway is being dysregulated based on the, up and down microRNAs and the genes it might suppress. So anyways, a tool that would use that. And overall, what I want to focus on are the pathways behaving the same way from the SGA, meaning the birth defects, and versus the GeneLab simulated space experiments. So that, and this is the mitochondrial related stuff. So the idea is, there's subtle effects happening in these pathways related, general pathways related globally with microRNAs. But in space with the microRNAs, we get a stronger response, meaning if you only need these kind of small changes, the mitochondrial or not mitochondrial, for the SGA to occur based on the microRNAs, imagine if they come back and have these huge elevated levels of change happening. So indeed, they'll have probably a greater impact. Potential birth, or the hypothesis would be there'd be a greater impact to all these pathways regulated to promote SGA if all you needed is this small increase in the pathway changes due to the microRNAs. And this is another way of looking at these overall pathways and how the relationship between the space flight, which is here on the x-axis, versus the SGA control, how similarly either up or down these pathways are predicted to be mitochondrial or non-mitochondrial related. So then, for a long story short and sake of time, I went in and said what microRNAs might be behaving or present in both the SGA datasets and the ImmPort datasets, and then to shorten it out, what micros are going the same exact direction between the datasets. So this is what these microRNAs here in this box popped out. So these microRNAs from the mouse, the SGA human data sets, and these are from the mice that were simulated space condition. You can see that there's only one microRNA that was down across this 146b, but the rest were pretty much up depending on the condition. Now, one thing you might want to say is, well, one's mice, one's humans. Are they related? And the answer is yes. Remember I said microRNAs are very highly conserved. So through my collaborator Paco, University of Lisbon, he did a homology type study, and this is what this is looking at. So one thing I missed about microRNA biology, when microRNAs, mature microRNAs, which all these are formed, they come from precursor microRNAs, which is from this little hair loop RNA, and then it's cleaved to form the mature microRNA that causes more of the damage and the functionality of it. So the precursor microRNAs might not be as conserved, but when he did the study, you could see that from the microRNAs that are in common between SGA and spaceflight, the majority of them are even 88% or 86%, the lowest conservation to even 100%, but all mature microRNAs that are here are 100% conserved between mice and humans. So that's amazing. So it's like, yes, anything that happens in the mice, if the micros pop up, the functionality will be the same in the humans. So when you look at what these 13 common microRNAs might be associated with function-wise, you can see that, and this is disease-wise first, sorry, disease-wise and then functional. I'll show you the next plot. You can see, on top of here, these microRNAs are indeed associated with birth and pregnancy issues, which is good to see. That means that we might be onto the right microRNAs. Now, a lot of microRNAs seem to be associated with cancer, but remember that remember these pathway databases that come up and these ways of analyzing it, cancer is most abundant for microRNA literature. So that's why some of these 13 microRNAs, maybe you see cancer popping up. But there's all other sorts of issues that pop up too, other than birth defects. And then what functionalities they have. I mean, there's mitochondrial. I tend to focus on that, which I'm not getting into in this talk. But, there's all these other issues that these microRNAs are related with functionally, from bone issues, CNS issues, cell cycle, and just different pathway databases using, looking at different responses that these microRNAs might have important implications in. So I'm not getting into too much details here of that. And then what we did was look at the targets. So, as I said, every microRNA can target hundreds of thousands of genes. So one thing we did is now look at across all 13 microRNAs, what are the common genes targeted by all 13? So that means those are probably the most heavily regulated genes by these 13. If 13 microns are targeting the genes, it would be the highly suppressed. There's about what was it? Oops, about 40 some gene targets. These are all the gene targets here. And those I said 10 or more common gene targets that would have out of the 13. So then what these genes are functionally behaving are, you can see things related on, mitochondrial things, unfolding protein response to, DNA response to metabolism of RNA, but they'll also be involved with many other functionality related with like immune functions like B cell activation and things like that. So there's a lot of factors really here with immune and development, as you might expect. So that's good to see that, we're on the right track for the targets. Now you say, how are these gene targets correlated with human-related issues? Remember, I mentioned the Inspiration4 data. So the Inspiration4 data, what we did, there was no microRNA sequencing done, unfortunately, but through the help of Chris and Zhang Kun is one of Chris's very talented scientists in the lab that he does a lot of work. He's like a superhero scientist, I call him. But what he did, since they had 2 females and 2 males, you could actually look at the differences of how the targets of these microRNAs will behave between the males and females. And if these microRNAs are being expressed in females more, and it's a female-dependent issue, maybe the targets, you might see some of these targeted genes higher suppression happening in the females versus the males. And indeed, depending on this is a chart here showing the gene targets. They did single-cell RNA-seq on the crew members here. And then we're looking at the gene targets related with each of these microRNAs, and whether the gene targets have been increased and the number of genes that have been increased or decreased. So you can see that indeed in the females, this is females versus male. So if it's increased like this, that means this higher proportion of it's being decreased in the blue in the female. So you can see, depending on the cell type, we do have a higher dependent female decrease of these genes than men. Depending on the cell type. So there's some cell type dependent factors happening. So this would give clues to be validated later. Maybe the microRNAs are behaving for the birth defects in certain cell types higher than in other cell types. But it is interesting to see indeed the gene targets that the microRNAs are targeting in the Inspiration4, that 3-day mission, which got exposed to high dose radiation. They definitely have a spike this is immediately after we return from flight. So this is R1. This is 45 and 82 days afterward. And you can see even depending on the cell type and the microRNA targets, you do have a higher suppression of these genes in men. So that was sort of indirect confirmation that maybe something's happening in humans. And for the sake of time, then we also looked at from other tissues from the simulated micro experiments, how these microRNAs are behaving. So these are circulating microRNAs. We're looking at the serum plasma, right? But how they get into other tissues. In those same mice experiments, we looked at heart and liver and muscle. And here in this top chart, we're looking at how the microRNAs might be expressed. Is it only in circulation? Do they get in other tissues? Depending on the tissue, they do seem to be expressed significantly in other tissues. We even looked at this twin study. That was this response to a reviewer's comment. The Scott Kelly Marquez men. So, the question was, how do you know if these microRNAs do exist really in females? And like, there is no actual astronaut data microRNA expressed in females. It's impossible to get. But let's see if they're expressed in anyone. And in the men, Scott Kelly versus Mark Kelly in the ground, you can see some of these microRNAs are indeed up compared to, post flight in the ground. But it's in the men. So, that's the closest thing you get, this response to a reviewer's question. And then we looked at a whole range of other GeneLab data sets in other tissues to see how the overall pattern of these gene targets are for the microRNA targets are for the genes. And then they can see a pattern here, which I'm not going to get into, but we see that. And overall, again, the microRNAs being increased or decreased in the different tissues. You can see kind of a pattern potentially. Like the liver, you can see some increase happening in the GCR versus like soleus muscle, you don't see as much happening. So you can say maybe due to the circulation and liver being sort of a filter, maybe that makes sense. These microRNAs you also might see that overexpression in the liver. So all this nice potential biomarkers, can there be a potential countermeasure now predicted that people go back in the lab and validate? So all these studies is all in silico. So this is all hypothesis generation. Something we have to do from this is now the actual models to run, experiments to validate the genes, and microRNAs, but while we're doing that, is there potential countermeasures? So with my collaborator Diego and other nice collaborators, but Diego's really the mastermind of this tool, the AI/ML tool we created. This paper just came out a couple of weeks ago, where it's an ML tool called SChemNet, where we basically use chemical similarity of small molecule drugs available on these small molecule drug databases to basically compare the chemical similarity and how microRNAs might be targeted by these small molecules. It's a tougher problem to crack, which he was able to do with. And the reason it is because the databases out there's a very small amount of data available comparing what molecules might target microRNAs. So I won't go into the details of this AI/ML tool, but basically the idea is now using his tool, we can predict potential small molecule drugs that could target the microRNAs. And in this paper, we actually ran validation experiments through our collaborators, Daniel and Dorothy and Rob Miller. And so we were able to actually predict from microRNAs what targets, small molecules we targeted. They bought them small molecule targets. They ran experiments in the lab, and indeed it worked. And we were able to mitigate, or as a therapeutic, we would say, oh, the damage was actually prevented. So it was nice. It's a nice model too. I'd be happy to collaborate with them on that. So we kind of use the same model here. We have our 13 microRNAs. So let's run Diego's tool, SChemNet, on the 13 microns and see what listed drugs pop up. So from all the molecules that pop out, there's a bunch of drugs that popped out, and you can see the overall mode of action that all these drugs might be behaving in different. Not all 13 microns appeared because some of them are not actually, you can't find any relationship to any small molecule drugs out there because the databases are lacking that information. So of course, these databases need to be further increased to include all microRNAs. But the microRNAs that do increase, you can see, for example, a lot of the drugs are targeting, depending on the microRNA, estrogen and female-related mode of action that drugs will target. Vitamin D is a big one. And other things and then the enriched drug indicators were like hyperthyroidism, which is also related to birth defects, things like that, menopause, and other things depending on the microRNA. Now, there's a bunch of, there's common drugs that these were predicted to target across the microRNAs. There's a bunch of other drugs, but we said, let's go down to the common drug. There's only 2 drugs that are commonly already approved by the FDA that were targeting by all the microRNAs. It was triamcinolone and parafluorodecolin. Now, these drugs you don't want to give to any pregnant woman, right? This is definitely you don't want to give to the pregnant woman. This is a potential countermeasure to maybe test as a way to keep the microRNA suppressed. Let's say if a female wants to go to space, and then take them off once they come back, so the microRNA is never expressed. Now, this is a glucocorticoid, and it could be a steroid kind of drug. Some you might not want to give that. And of course, everything here you have to validate and test. These are just suggestions and in silico predictions. So, this might be something you might think of. That might be potential. Now, parafluorodecolin, actually, there's some papers out that people have done looking at this. I mean, originally it was discovered in the 80s as a way for liquid breathing and applied topically for extra oxygen. But for preterm infants and studies that they've done that actually have shown that has maybe potential help in like LAM studies and other studies they've done to reduce the chance of preterm infants and maybe help. Nothing directly with SGA, but might have some influence there. Again, all this stuff is just suggestions. It's in silico and you have to be validated. But the beauty is with this large network of people working on it, some people from GeneLab, as you can see here, folks, folks from all around the world were able to help and really get a nice paper out to help predict a potential microRNA signature and potential therapeutic that we could now go back in the lab and all apply for grants and then go back in the lab and test and see if it actually is working and is true. So, and another thing, it's this future studies too, that one thing that NASA, and we know it said you got to go to the moon. Now, when you're on the moon, you have this thing called regolith, the moon dust. And in this grant we're testing like the regolith about looking at liver issues and other factors there, really mitochondrial and microRNA driven issues. But also to think about, when astronauts, as we know, the regolith is very powdery and sticky. So the issue is when they take their suits off and they get back in from the moon, it gets in the atmosphere and they breathe it. No one knows what biological effects it'll have. And my question would be, does regolith impact birth issues too, like SGA. That could be another impact of maybe people here could work with me to figure out. But that's it. Okay, hopefully, I think I have 5 minutes, but now just questions. I'll stop sharing. Thank you for listening.
What a great work. Any questions? Okay, while people are posting questions, I do have a question. It's so interesting. So you did mention earlier that you were looking at the data. You found the data in ImmPort, you found the data in GeneLab. So your hypothesis was in mind for a long time, and then you happened to find the data. How did that work?
So I didn't have a hypothesis. I went in thinking, what data set can I use? I was kind of like hypothesis agnostic first. I was like, I wanted to see what data is in ImmPort. And then once the data, I could see the data, I could say then what hypothesis I could generate. Because if I had a specific hypothesis in mind and go to ImmPort, the data set might not have been there. You know, the creativity part is the key for combining these two data set platform, because as you're probably aware, it's not as easy for all conditions. Like I know ImmPort has a lot of virology type data, and one thing that maybe people get help with. I mean, the virus. I was first looking at that point because we know some viruses do reactivate in space, like the herpes complex viruses or chickenpox. So that's an issue, or EBV viruses a potential reactivated. So I was trying to find that, but it's harder for me to find data that might be relatable to GeneLab. So then eventually I stumbled upon this one as like microRNAs, and then once I saw this database, then my hypothesis generated because I knew I had my microRNA data set already made. So that's how the train of thought happened. I didn't have actually, because and I already knew, for example, female reproductive issues is a big concern and it's understudied in space. So I know that was a good hypothesis to pursue. But that's just one of many that people can pursue. If someone already has a hypothesis in mind, I mean, that's even better because then you could look at ImmPort and then find a potential data set they could get. But the tough thing is finding a data set that's compatible with GeneLab. And that's just creativity and being open to trying to be indirect or, be flexible, I guess.
This is a great example of how the data are used, because our lab also does a lot of data reuse projects. So when we go to ImmPort, we look at, okay, what data is available there and how creative you could be and ask some scientific questions, get a scientific insight into it, right? Probably I might not have thought about it, but now the data sets are already available. So let's do some meta-analysis, combine the data sets and be creative. Thank you.
Exactly.
Sanchita, I have one additional question here for Afshin. What advice would you give to someone who is maybe exploring open data and data reuse for the first time? Do you have any tips or advice you'd give?
I would say just be as creative as you can be. I mean, and I mean, as I just mentioned to Sanchita's question, is that don't be the focus, don't be narrow, right? I guess be open and don't be afraid. Like my specialty is not female reproductive biology, but make sure you have a Rolodex of a lot of people in your network to bring in to help you out, because once you have, let's say, a data set outside your expertise, you might have the computational skills to analyze the data, but the interpretation, you need your experts to come in. Like, for example, one of the authors in our paper is Jean, which is she's a gynecologist, and she also does a lot of research in female reproductive systems. So she was able to really help us interpret some of the data in the discussion section and add a lot to that. So that helped. So that's the part where I think when you don't have experience, when you're mining the data, one is just take your time and look at the databases, because that takes a while too. Like, opens the GeneLab Open Science Data Platform is pretty slick. It's easy to go, but you still need to familiarize yourself with what's there. Same with ImmPort. You need to know what's there and how to navigate the system. And after that, just start putting keywords in. Let's say if you read an article on, some infectious disease, obviously you want to look at that in ImmPort first, and then think of how could that relate with, as I just said, reactivation of viruses in space, and see if any data sets are available on GeneLab. Or even if there are no data sets available on GeneLab, think about what could be, let's say, the organ that let's say a virus might get reactivated, like a spleen or immune-related functional organ, and then find the pattern of genes that might be dysregulated due to the infection and see if that pattern of genes are apparent in space. So that's an indirect correlation, but maybe that's good enough to come up with a creative way to come up with a paper.
Thank you so much.
Great. I think we are around the top of the hour, so, and I don't see any questions under Q&A. Okay, so thanks again, Dr. Beshari, for this very nice presentation. And I would also like to thank the participants for joining today's webinar. I did post under the chat that all the webinar recordings, slides, and additional materials are available for download and viewing on the ImmPort website. And if you want to stay connected to know more about the new data sets that's coming in, so you can stay connected through subscribing to our newsletter and then going on to the website and join our mailing list. So we hope to see you for the next one. And thank you again for your participation.
Thanks.